My name is Ioana Iorga. I'm Business Development Manager, Health and Personal Care at NIZO.
And I'm Janneke Ouwerkerk, Group Leader Food Safety and Fermentation at NIZO.
The fields of probiotics and microbiome are very important to NIZO's activities, so we are pleased to share our own perspectives and learnings in this area. The market for probiotics and live biotherapeutic products continues to grow as we increase our understanding of how they can improve health. Probiotic and pharma companies are looking for ways to use the naturally occurring bacteria in our bodies to amend dysbiosis in the gut, skin, and more. Then they must get their products to market quickly and cost-effectively. But there are many challenges that will define a company's success or failure, especially when working with living organisms that don't always behave as we want. With our science-based expertise in probiotic screening, optimization, and production, we partner with companies from small startups to major multinationals. We support them along their probiotics journey with a multi-stage approach that includes discovery of microbiome modulators, process development and upscaling, food-grade production or tech transfer, and health benefit substantiation. And above all, we share with them our passion for probiotics. The global probiotics and microbiome therapeutics markets offer exciting opportunities. The rise in digestive health diseases and chronic skin problems is driving demand for probiotics and live biotherapeutic products. Consumers also want more functional foods and beverages containing probiotics as they become increasingly aware of the potential benefits. And there is a growing evidence that some probiotics and live biotherapeutic products may improve the efficacy of certain medical treatments, in oncology, for example. As a result, in 2019, the global probiotics market reached $44.2 billion, and it is expected to grow strongly with a forecast compound annual growth rate of 7.7% between 2020 and 2025. The global microbiome therapeutics market is reaching new heights as well. In 2019, it was worth $42 million, and it is expected to grow 42.8% between 2020 and 2025. But the probiotics universe is complex. Bacteria can be highly specialized, filling specific niches. Novel strains may not be well characterized, and available literature can be limited. Furthermore, a bacterium may require a specific medium to thrive. Externally, legislation regarding bacterial safety levels impacts how bacteria may be studied and in which types of products they may be used. NIZO has helped many companies to identify, grow, optimize, and substantiate the health benefits of probiotics and live biotherapeutic products. We have carried out hundreds of fermentation and media optimization projects, as well as a broad range of strict anaerobic productions and tech transfers and successful clinical trials. With over 100 professionals, a total of 42 laboratories, including general, BSL-1, BSL-2, and food grade, and our food grade pilot plant, we can provide customers with a broad range of services and expertise. As an independent research company, our focus is fully on the customer's project. Importantly, the intellectual property from the project remains completely in the customer's ownership, as agreed in our project proposals.
To maximize the value of our in-house expertise, we have developed robust science-based methods that provide a knowledge-driven selection and assessment of bacterial strains, optimized production of microorganisms, and investigation and substantiating of the health benefits. Discovery starts in silico. The bacterial genome already holds a wealth of information that can be used to predict the strain's behavior and potential use as a probiotic product. Combining our bioinformatics expertise, custom-designed functional modules, and expert interpretation, we support our customers in selecting the most suitable and promising strains for probiotics. There are two approaches for which NIZO's in silico expertise provides high value to customers seeking to develop a functional product. In the first case, the customer has focused on a specific group of bacterial species or strains. The beneficial and unique probiotic properties are explored and identified before selecting in vitro substantiation assays or moving to production. On the other hand, the focus may rather be on a specific functionality of interest, such as reducing acne or easing constipation in the gut. In this case, a pool of bacterial genomes is screened for genes that support the desired health aspects. The number of possible contenders is then reduced to select the most promising strains for extensive in vitro trials. Once the strain has been selected as promising for use as probiotic or live biotherapeutic product, NIZO supports the manufacturer with input for required strain safety documentation. This includes data on the presence of any genes that could compromise safety, such as antibiotic resistance or virulence factors. But then we go further, interpreting the data based on evolutionary relationships with other strains and the potential transferability of the genomic properties. We can also validate the in silico results with additional in vitro studies. And we can help with preparing a dossier on downgrading a bacterium from BSL-2 to BSL-1, which is critical for any bacterium that will be used in a food or beverage product in Europe. With the initial selection complete, the next step is designing a process to produce viable bacteria in large quantities. As living organisms, bacteria respond to their environment. Optimizing their production is both a science and an art. Bacteria can be very hard to grow, especially strict anaerobic bacteria that come from the zero oxygen environment of the gut. Other bacteria grow slowly, perhaps too slow to make them a good choice for manufacturing. Some are easy to grow but difficult to keep alive, which could impact the yield, shelf life and thereby cost effectiveness of the end product. In any case, the bacterial production process must be reproducible and scalable. Each of these potential challenges is impacted by both the bacteria itself and by the process used. NIZO's approach takes both aspects into account, using our probiotic optimization pipeline. The probiotic optimization pipeline starts with confirming the identity and purity of the bacterial strain that has been selected. Still at the micro level, using high throughput screening, we employ up to 3,000 growth conditions for mainly the upstream processing within a time frame of weeks. With the vast amount of data collected, automated colony forming unit plating and counting is a necessary tool. To optimize the selected growth conditions, we then upscale to half-liter production. In these first high and medium throughput screenings, we explore interaction effects of various medium components, pHs and cryoprotectants. We also test the optimal growth phase for harvesting. We can screen up to 12 fermentation conditions at the same time and test freeze-drying or spray-drying formulations at laboratory scale. Upscaling to 40 liter and 600 liters, we test flexible downstream options, such as centrifugation and cross-flow filtration, formulations such as liquid powder and capsules, and accelerated shelf life conditions. Once the conditions are optimized, we can move up to full anaerobic scaling from 40 liters to 600 liters. At this point, for biotherapeutics, we can carry out the tech transfer to the pharmaceutical contract manufacturing organization. For probiotics, however, we can take the process even further. In our food-grade pilot plant, which has 600 liter and 4,500 liter scale equipment, we can manufacture formulations for food-grade and cosmetic-grade production. This step-by-step upscaling is critical because even small changes can have a big impact on how the bacterium reacts. As part of NIZO's strategic innovation project, we optimized the upstream conditions of Faecalibacterium prasnitzi. This strict anaerobe is notoriously difficult to grow. The half-liter production was run twice independently, controlling pH and temperature while ensuring an anaerobic environment. This graph clearly illustrates the reproducibility of the developed process. The process was then successfully upscaled to 40 liters, showing only minor deviations compared to the half-liter run. This research confirmed the successful reproducibility of the process for this very challenging bacterium. And it demonstrated how a step approach to upscaling supports a robust production process. During the upscale, enough material is produced to begin in vitro testing of bioactivity, a time and cost-effective step in screening for potential health benefits of the bacterium. Using in vitro testing, NIZO can provide robust insight to the customer to narrow down which bacterial strains are most promising for additional, more costly clinical trials. NIZO's in vitro studies can reveal not only what functionalities a bacterium may offer, but also the mechanism of action, which is a critical part of building a successful health claim dossier. These studies may also reveal new functionalities, further enriching the story for the customer's product. NIZO offers a range of in vitro models, which provide a simplified version of specific human biological niches, such as the skin or gut. Microcolon and microskin are NIZO's unique microbial community models. They provide micro-representations of the skin or gut. Using samples from selected volunteers from the target population, we can carry out high-throughput, cost-effective screening of modulations of the microbiota composition and function. NIZO's cell-based medium to high throughput screening assays use a consistent and reproducible approach to evaluate specific functionalities of probiotics and dietary supplements that could be helpful to predict effects in humans. Human or animal gut or skin epithelial cell models are used to explore effects of probiotics on barrier function, pathogen adhesion and modulation of local inflammatory response. Immune cell models provide insight into immune function and modulation of systemic inflammation. For all in vitro studies, NIZO works directly with the customer to discuss topics, approaches and areas of interest. A customized approach is created, which may include a combination of cell lines and microbial models.
It's clear that bringing to the market products containing living organisms, such as probiotics and microbiome therapeutics, involves a multitude of challenges. But with a robust, scientific and multidisciplinary knowledge-based approach, companies can employ a stepwise methodology to fast-track their development and entry into the clinical trial phase. Start with a targeted approach. Combine a state-of-the-art bioinformatics approach with expert interpretation of generated data to screen for the genomic potential for specific functionalities or for properties that may compromise safety. Select the most promising strains for probiotic products and then validate the in silico predictions. Optimize the medium composition, harvesting moment, and formulation to enable a robust, upscalable process. Screen and test food or pharma-grade ingredients early in the research and development to find the best compilation of ingredients. This ensures you are using the best medium from the very start of the process. For microbial therapeutics, consider the needs of the tech transfer to the pharmaceutical contract manufacturing organization from the beginning to guarantee a smooth and robust handover. For downstream processing, keep in mind new formulation technologies that are being developed, such as spray chilling and electrospinning. Make sure to also include the application and shelf life challenges at an early stage. Finally, in vitro studies offer a fast, cost-effective way to screen your bacterial candidates, so you are bringing only the most promising to clinical trials. Assess and develop a greater understanding of the functionalities of the strains, using models that mimic human biological niches. The microbiome ecosystem is vast and complex, but building on proven expertise and science-driven models and methods, NIZO offers a pipeline that can support you from discovery through optimization to health benefit substantiation, even for the most difficult, strict anaerobic bacteria. We would be happy to discuss with you in more detail how NIZO can support you in your own probiotic journey. Contact us for more information.